Hi, this is Miss Litton, and these are my wonderful AP biologists in 7th period review. Say hi.
Hi.
Okay, we already did an AP Unit 3 essay review, which was recorded and posted up on YouTube. I just want you to know what was in it, okay? We discussed already about these different essays about mitosis and meiosis, so I want to encourage you to look at that. We talked about ways that we have variety. We talked about cancer and the failure of genetic control. That is on the review. Who was here at that review? Who has watched it already? Oh, look at you, smarty McSmartest. Good. Okay. We talked about Mendel's conclusions and how things can deviate from Mendel's conclusions. We also talked about gene interactions and examples of each of those and how that plays out. And then that brought us to this question right here. Experiments by the following scientists provided critical information concerning DNA. Briefly describe each classical experiment and indicate how it provided evidence for the chemical nature of the gene. Okay, so the first one is Hershey and Chase. Okay, so Hershey and Chase, because we always chase down what? Hershey's chocolate. Excellent. Okay, so what was Hershey and Chase about? What were we using? Yes? Bacteriophage, which we now know, okay, the structure is, oh, weird. Why am I jumping around? Weirdness. Yeah, I think I need to recalibrate. Let me do that real quick before I go nuts. Okay, either that or I need to dust my board. Okay, bacteriophage, and they labeled the protein with what? What did they label the protein with?
Salt.
No, it's still jumping around. It's going to drive me crazy. What did they label the protein with? Well, there's P32, but that's not what they labeled that. Yes?
S35.
S35. Whoa, okay. Wherever I write, it's moving it. And then inside, they labeled the nucleic acid with what? Ah, P32, good. And the point of this experiment was they were trying to understand, they knew viruses overtook bacteria and made those bacteria start to generate more viruses. And what they're wondering is, what is that transforming agent? Is it a protein or is it a nucleic acid? And remember, they couldn't see it. They didn't have electron microscopy, so they had no idea where the protein or where the nucleic acid was. They just knew a virus had both of those things. So when they allowed the bacteriophage to infect the bacterium, and then they separated the phage from the bacterium, how did they separate it? Using a blender. And then they did what? Centrifuge. Heavier elements go to the bottom when you do a centrifuge, and bacteria weigh more than the virus. So they were looking to see what's down there with the bacteria. Is it P32 or is it S35? Okay, and since it was P32 down there, they knew what the virus had put into the bacteria was a nucleic acid. Okay, clarifying questions on that. Who firmed that up? Avery what? Oh, sorry, move on. Never mind. Sorry, I was skipping an experiment. Okay, any questions on that one? Okay, moving on to the second one. Griffith. Now, when I think Griffith, I think what? Griffith Park, and then I think mice. Okay? So he had two bacteria, one which was lethal to the mice. Okay, there was S and there was R, and make sure you know the difference between those two. S is bad, exactly. S is bad. It had a smooth capsule on it, that's why they called it S. And so he knew if he injected S into the mice, of the bacteria, that the mice would what? S causes them to die. But if he injected R, they would what? Live. So what did he do to the S? He killed it and then injected into the mice, and the mice would what? Live. So the S killed by heat, okay, definitely did not make the mice sick. But then he took the S that was killed and mixed it with R that was alive, the stuff that didn't normally. And the mice were still what? Dying. And when he analyzed their bodies and re-looked, sure enough, it was like S had come alive again. And it wasn't that S had come alive, R got something from S. And it got pieces of its genetic code, which transformed the R non-lethal into being virulent, into being lethal. So it got that genetic code. And they knew that because Avery, McLeod, and McCarty, the follow-up experimenters, what they were able to tell is that when they digested DNA, that transformation didn't take place. But when they digested RNA and proteins, the transformation still took place. So they knew it was the DNA that was the important part. That's Avery, McLeod, and McCarty. Now, Messelson and Stahl, okay? I'm going to mention them, okay? In your brain, absorb them. Do you see them? And I think I misspelled Messelson, to be honest with you. I think it has two S's. There's an S here and an S there, and that makes me think about DNA, like this, Messelson and Stahl, and I think the S, like when you make it. What they did is Watson and Crick came up with the idea of semi-conservative replication, that the DNA would unwind and unzip and each strand would serve as a template for a new strand. Messelson and Stahl did the experiment to support their hypothesis. And again, used an isotope and basically grew up, there was heavy nitrogen in the experiment, and they grew them up in heavy nitrogen and then replaced it with light nitrogen. And after one replication, it was a mixture of, it was one heavy strand and one light strand. Now, I'm telling it to you only because it's not one of their scientists on the list, but it makes me nervous because sometimes they say things aren't going to happen and then they end up happening anyway. I'm telling you, I'm not asking you about it, okay? But I want you to be aware of it. And you can read about that experiment in your book, okay? Any clarifying questions on this essay? Yes?
Which one are you asking us about?
Messelson and
Okay.
Stahl. Yeah. Yes?
Are Messelson and Stahl also the people that made the amino acid chart?
No, that's Nuremberg and Matad. Sorry, I like to say the names like that. Okay. Any other questions on that? All right. So let's move on to the next one. It says describe the biochemical composition. So what is DNA made out of? And its structure. How is it arranged? And replication of DNA. And be sure to include any enzymes that are required. Now, if you're going to talk about that, I'm going to think there's some scientists that are going to come to mind. Who are some scientists that are going to come to mind? Watson and Crick ultimately, but who else? Historically, what other scientists? Chargaff. What did Chargaff tell you? The number of adenines you had was the same as the number of thymines, though the number of adenines and thymines could vary in any different species. A's and T's were about the same and G's and C's were about the same. Did he know what that meant? No. Okay, but at least that's, you want to attribute that to Chargaff. Then who was another contributor? Rosalind Franklin and Maurice Wilkins, who they all, you know. And what did they do? They would shine what? X-rays through crystallized DNA. And when you analyze those X-rays, people who know how to do that, okay, saw kind of an X in the center, right? And that showed us that it was a helix, and then they could also tell it was the same diameter. Remember our numbers for diameter, 2, 3, 4, right? 2 nanometers across, 3.4 nanometers per turn, and 0.34 between bases. Remember those numbers? Sure, 2, 3, 4. DNA is number 1. All right, now, you are going to want to be able to make a simple diagram of DNA. And I'm going to tell you it's usually easier to start with your bases first and then add your phosphates and sugars because you tend to mess up if you do it the other way. Okay, and of course with my board not being in alignment, this is going to be super fun. Okay, but I would draw like a bigger one like this, and this would, let's say, be adenine. Okay, and then I would go ... Okay, and then it is bound to who? Thymine. Okay, why did I draw it like that not so well? Because I was stressing it. What? Yeah, adenine is a purine and thymine is a pyrimidine. So it's smaller, right? And then I could do, if I wanted to, I could do a guan. Oh my gosh. We could make this guanine, and then we could make this one over here. We could make it what? Cytosine. Now, I would draw 3 hydrogen bonds because that's what it has. Okay? And again, this drawing is super hideous. Okay, but my board just is freaking out. You would want to identify that these are hydrogen bonds. You would want to identify things like purines and pyrimidines. Okay, all of that would happen. Okay, now what are these bases connected to? What are they? Sugars, right? So you don't have to be, oh my gosh. Okay? It's like a blind person drawing. Okay. Now notice I'm trying to make them go in 2 different ways, right? Do you see that? Okay. And remember we said this would be carbon number here, where I'm actually pointing on the board. This would be what? 1, 2, 3, 4, and then here would be 5. And this is where your phosphate would be, that's then connected to your next sugar. Okay? And here, okay, actually I should have looked a little carefully. 1, 2, 3, 4, 5, and then this would be connected to my phosphate, which is connected here. That would be a better one. Oh my gosh. Okay, and then I would have to label this as what? What is this thing? Yeah. Oh my gosh. Deoxyribose, and then this is phosphate. Okay, I can't even write. And then I would list the bases and what they are. Oh my gosh, I can't believe you're taking pictures of this hideous thing. It would be better to Google hand-drawn DNA, okay, and put that in your notes rather than my board out of alignment. Okay? Now, the next thing we need to talk about is the replication of DNA. So if we were going to make a list of things and enzymes we needed to include in the replication of DNA, what would we include first? Yes?
Topoisomerase.
What? Topoisomerase. And we would tell what topoisomerase does. Then who would we talk about? Helicase. Then who would we? SSB. Good. Then who would we include?
DNA polymerase.
What, first we'd have to do what? RNA primase. Now, it's here that you would be differentiating between leading and lagging strands, right? Or you could say something to the effect that RNA primase is laid down one time on the leading strand, but multiple times on the lagging strands because the sugars are not in the appropriate orientation. Okay, but you would have to say something about that, okay? So you have to say something about leading, let me just not look, and lagging. Okay, at that point. Okay, now who am I going to mention? DNA what? Polymerase 3. Okay, and you would talk about how it works off of the primer that the RNA primase lays down. And you would say things like bringing in complementary nucleotides. You know, and you would mention to me, adenine is complementary to thymine, thymine to adenine, guanine to cytosine. So you would explain to me about the different complementary nucleotides. Who would you need to talk about next? What? DNA poly 1, and you would talk about how it removes the primer and replaces it with the appropriate DNA nucleotide. Okay, and then who would you need to talk about next? Ligase. And you would tell me how ligase seals the phosphate sugar backbone of the areas where the primer was removed and DNA was put in its place. Okay, because DNA polymerase is going to catalyze the building of that DNA as you're going through. It's just wherever you took out primer that you're going to need to use the ligase. Okay, clarifying questions on that? Yes?
Can you explain the meaning of ligase?
Sure. Okay. So when you are at, let me see if I just switch this, if this does anything. If I go around one time. How about now? No. Let me see if I need to dust my board. How about now? No. Okay, all right. So on your leading and your lagging strand, okay, when your DNA becomes unzipped, okay, and let's say we're going to start at this point right here. If right here, this is 3', then right here would have to be what? 5. It's the other side, right? And if here, that means 5' is here and 3' is here. Do you agree with that? Okay, so I need to put down a little bit of primer because it reads 3 to 5, making complementary what? Complementary 5 to 3. So what I would do is I would lay a little bit of primer here and a little bit of primer here. And then I'm going to change to green, my new strand of DNA that's complementary. It could read right off of this, making complementary, and read right off of this and make complementary DNA. 3 to 5, wherever you start at that point, like you're at an intersection, this way or this way, right? On this one, I can't read this. So what I'm going to do is I'm going to lay a little primer here and then work off of this. And then a little primer here, and then I'm going to work off of this. Oh, work off of this. And then I'm going to put a little primer here, and then I'm going to work off of this. All right? And then what I have to do is I need to erase, erase, and I'm going to remove the primer, right? And then I'm going to use DNA polymerase 1, who's going to replace that primer with DNA. And I'm going to do that again. Remove my primer and replace it with DNA. And remove my primer, and replace it with DNA. And then ligase is going to seal those phosphate sugar bonds to keep it all together. And, can I do a little bit more? So on this side, this would be my lagging strand. So I'm going to go here and put my primer down, and then I'm going to go again. Damn it. I'm glad I recorded my swearing. Okay, and I'm going to put down a little more primer and then lay down my DNA. And these right here, my friends, those are called your Okazaki fragments. And they are about 1,000 to 2,000 base pairs long, and these primers are about 10 base pairs. Does that help?
Yeah.
Okay. Questions?
This all occurs during like the DNA polymerase, right?
The removing of the primer, is that what you're asking?
No, just like.
All of this is happening during the S stage of interphase. And now these, my friends, are what? Sister chromatids. Okay, so all of these, right? These are going to be sister chromatids held together by a centromere, which separate during anaphase. Okay, put it together. Clarifying questions. Are you in the Shadowlands about anything? Yes.
So when you're copying it,
I can't hear. Shh, go.
when you're copying it, you go from 3 to 5, right?
Reading 3 to 5, making 5 to 3, because it's always, right, anti-parallel. Yeah. What else? Okay, so where I've shown you this before, okay, is we were looking at just one strand like this, and we were saying, okay, if this is 3', then this is 5. We were just looking at 1. But see, this continues over here, and this is what's called then the replication bubble. So we were just looking at one edge just to learn it, but it really would happen in the middle of something. Does that make sense? All good? We were just looking at 1/2 of the replication bubble when we were looking here, and we're saying, oh, this is 3' in, we just lay down a little primer here, and we're gonna work off, right? That's the leading strand. Over here, we have to lay down primer multiple times, and then work off of it. Okay, more on that. Okay. Then the last question said, describe the steps of protein synthesis beginning with transcription and ending with the release of a polypeptide from the ribosome. Include in your answer discussion of how the different types of RNA function in this process. So what would be the first thing we needed to mention? Topo and HeLa, right? And then we're going to use what? RNA polymerase to make what? mRNA. And this is where you could say messenger RNA, making a copy of the DNA. So RNA polymerase will bring in complementary RNA nucleotides. RNA is different from DNA. It has the sugar ribose. Wherever you would have a thymine, instead you have a uracil. RNA is single-stranded, not double-stranded. Okay? Now, once we've made the copy, okay, mRNA, okay, it's going to detach from the DNA, and the DNA is going to zip back up, yes? Okay, now you would have a processing time. So you have your primary transcript of RNA, and you have to make your mature RNA. So what are all the things that have to happen during processing? You have to put on a cap, 7-methylguanosine. You have to put on a tail, 100 to 200 adenines, and you have to cut out the introns, leave the nucleus. So this is the processing that has to take place. This process right here, what did we just describe?
Transcription.
This is transcription. Okay, that is transcription, going from DNA to what? RNA. Messenger RNA will be translated, right? The messenger RNA will be translated. If the RNA we make is rRNA, it will go and be a component of a ribosome. If it is tRNA, it will do the job of transferring amino acids to a ribosome, right? Just the mRNA will actually be translated. Questions on transcription? Okay? Are you sure? Ask the person next to you, because I'm here right now, so now's the time to ask me these things. Yeah?
What does it say after the word processing?
Oh, primary transcript to mature DNA. Yeah, I can't read it either because the board's not in alignment. I can't even write. That would be primary transcript becoming mature. Oh, it should be mature RNA. Sorry, mature RNA. Anything else just on transcription? Okay, yes?
Well, when it first separates, so when the DNA is separated by the topoisomerase and helicase, it then becomes RNA, or is it?
It doesn't become. Okay, very good to just say that. Nothing becomes. It doesn't, become something other than what it was. You're using it as a template to generate RNA. Okay, does that make sense to you? It is a template. So this is how do I build my RNA? I look and do what the DNA tells me. The DNA says adenine, I bring in a uracil. If the DNA says guanine, I bring in a cytosine, because that is complementary. Okay? More questions along that line? Yes?
After translation, when the tRNA jumps off its
Okay, I don't wanna jump into that, I wanna answer this question. Anything else on transcription? Okay, moving on then to translation. Okay? Translation. Okay, so when we do a translation, the steps you would want to include is that the what? mRNA hooks up with a small, then large ribosomal subunits. Okay, I would want to mention about what a codon is, which is every what? 3 bases. And then you'd want to say what the codon codes for. Most code for most possible codons that you could generate. Code for what? What do they code for? Amino acids. Okay. You have some stop codons. And you have one initiation or start codon, right? Which happens to be AUG. That's universal. And AUG codes for methionine every single time. Okay? Then you want to say that tRNA, right, has anticodon that is complementary to the what? Codon at its feet, and what is it carrying on its head? What? And it also, transfers an amino acid. On its head, it has an amino acid. On its feet, it has an anticodon that is complementary to the codon. tRNA1 lands, tRNA2 lands, tRNA1 gives his amino acid to what? tRNA2. Who facilitates the process? Ribosome, because it is a ribosome. And then tRNA1 leaves. tRNA3 lands, right? tRNA2 puts his amino acid, right? Amino acids. And this continues until you reach a stop codon. Now, what you might want to do is up in your, when you talk about the small and large ribosomal subunit, is mention it about the 3 spots. If you go back up in your notes to where you have, right? What are the 3 spots on the ribosome? Yeah, A, P, and E. A is where just the next tRNA lands. P is the one that already has the protein attached to it. And E is, yep. Yes.
So 2 things.
Listen.
The first tRNA, it lands in the P spot?
Yes, the very first one lands in the P spot.
Okay, and what happens to the like, so it dumps off its amino acid, then it leaves to the E site. What happens to it afterwards?
Okay, now keep in mind, it's actually, it itself is never changing. It's the ribosome and where it is in the process. The ribosome moves. So I could have 2 tRNAs right here, right? I'm in 2 codons right here. If I'm a ribosome, right, I'm shifting where he might have been in the P spot, now he's in the exit spot, now he's leaving. And the next guy here is in the A spot, now he's in the P site, and I'm shifting. You guys are not moving. mRNA is staying put, the ribosome shifts.
Yep.
So it shifts the tRNA into a new spot. Ask your question again.
But once the tRNA
Guys, don't talk when he's asking his question. Go.
Once the tRNA leaves.
Oh, then you're going to need an enzyme who's going to add on another amino acid to that particular tRNA.
Okay.
Yes.
So the codon is every 3 base pairs on the mRNA.
Not base pairs, because there are no base pairs. Yeah.
And what exactly?
You could just say bases.
Bases on. And what exactly does ribosome do?
A ribosome, on the ribosome, you know how you have to get that peptide bond, do a dehydration synthesis to join those amino acids together? The ribosome facilitates that process, the ribosome. You're just saying it's an enzyme too. It's a workbench and an enzyme. Okay, what else? Okay, those are all the essays. Keep in mind, can I ask you other things? Yes. Okay, I can ask you other things. These are just ones that you can prepare for that I more than likely might ask one or two of, but you need to be ready to understand other things, which if you're doing a good job writing all your essays, if you're doing a good job processing information, then you should be able to do okay on those other ones. Now I'm going to tell you right here, just so you know, the essays when you take a team task are the longest part of it. Because it's not like a multiple choice where the answer is there and you're selecting one. You have to come up with the words and the vocabulary to express your culminating collaborative thoughts, right? So what I want to do that's a little different this time, I want to take the multiple choice first, do it quickly, get it done, so we have the majority of the time ready for the essay, okay? That will allow you to take the pressure off, and then you'll know you can work till the end, okay? So we'll do the multiple choice a little bit quicker, and then let me give you a very strong suggestion. When you do this, and I will make the suggestion tomorrow as well, you will each get your own copy of the multiple choice test. You will each have your own answer sheet. Plow through all 25 of the questions, answering what you think is the right answer. Click them in the whole 9 yards, okay? Or at least, at the very least, write them down on your paper. Once, if we're partners, okay, once she and I are both done, I'm going to be like, good. You know, I might be going back to visit a couple that I had questions or circled. She and I will then compare papers. Only focus on the ones we disagree on, right? Only focus on the ones we disagree on. Hammer out those. If she and I still disagree and I say 10 is C and she says 10 is A, she can put whatever she wants down, right? She has her own clicker. So she's not going to take my clicker and go, okay? I can put what I want, she can put what she wants. But focus on those. Then if you want to take the time to go back and reread everything, but chances are if both of you agreed on an answer, it's probably right. Could you have made a mistake? Sure. Any ones that you think maybe I moved too fast on that one, circle those. If you really are unsure, circle and star it. But put what you think your answer is. Come up with a think, because otherwise if she puts nothing down and she's wavering between A and C and I come up on her and go, you know it's C. Oh my gosh, you're so stupid. Okay? That's going to make her go, okay, I guess it's C. Put what you think it is. Okay? Then you can have a better discussion. Commit to your answers. When we're done with the multiple choice, then you'll have the rest of the time for the essay. The way I photocopied the essay, instead of front and back, they're on 2 different sheets. That way one of you can work on one and one of you can work on the other. Okay? And what you're going to want to do is you'll have your own scratch paper like we always do. Talk through first. Hey, what do we think we want to write for each of these? Jot down your key points down on your notebook paper and then start writing them down into the actual answer sheet. Okay? After that. Yes?
Is the highly suggested reading and thinking going to be on?
That's a possibility. It's always a possibility. Yes?
So like you said with the scratch paper, is there only going to be one piece, or each person in the group will have their own piece of scratch paper?
You would have your own piece of scratch paper, right? Yeah. See if your brother will loan you a piece. I don't know. Maybe, maybe not.
I'm a nice guy.
You are. Wait, why are children coughing? Okay, go ahead. Yeah, we can do all songs. What are the songs? Mitosis, meiosis, right? Okay, and do you know all the songs are online too? Did you know that? They're all recorded. Do you still want me to go over them with you? Okay, all right. Do you want me to stop the recording here since the songs are online? Okay, remember if it's really late at night, have a piece of toast.